everyone joining. Sorry about the technical issues. We didn't realize people were on when it looks like we have 35 participants on the call. And so the work that Adam Gordon is going to present today is on behalf of the Coordinating Center and the Sequencing Centers. And this is based on our attempt, ours being the network, to putting together the target panel for sequencing. And so the initial requests from the NHGRI were for sites to list your 100 highest priority genes plus a second list of 25 genes, and then a list of the top 5 genes, and ranking an additional 5 genes if we do decide to go with the ACMG list. And so we purposely did not send the matrix out just so we could really demonstrate on this call and just kind of show you the content. And then this will be distributed immediately after the call. And we want you and your sites to really peruse the matrix and to provide feedback in an effort for next week's PI call. And with that, I'll turn this over to Adam Gordon, and he will start with a short presentation and then demonstrate the Excel matrix itself. So Adam, go ahead and take over.
Hi, thanks, David. So for those of you I haven't met, my name's Adam Gordon. I'm a postdoc here at UW with the Coordinating Center in Medical Genetics. And I've been sort of tasked with collating and putting together all the gene requests from all the sites for the new eMERGE 3 sequencing panel, as well as getting sort of ready in terms of thinking about how to put it all together. My experience in doing this sort of thing is through a panel known as PGRN-seq, which we went through in the pharmacogenetics network, a very similar process of gene requests for a custom capture panel, which I was involved in. And so hopefully I think I can get this process going pretty efficiently and pretty pragmatically based on what we experienced with the PGRN project before. I also wanted to say that this,
could you move closer to the phone so we could hear you a little bit clearer, please?
Oh, sure. Yeah, absolutely. And I was going to say, please feel free to interrupt me at any point with stuff like that.
Whatever you did was worse. Try again.
Sorry? That was worse? Better?
Yeah, that's better. Thanks.
Okay, yeah. My mic is probably strange. All right, yeah, speak up if it's going quiet again. But all right, just to move right into the slides here. So as David already told you, the initial request to the sites was from the NHGRI, list of 100 genes plus second priority list of 25. And then from the eMERGE PIs, a list of the top 5 genes, followed by an additional +5 if you're interested in. So when I put these together from all 9 sites, we ended up with 494 different genes that were requested overall. That's 494 uniques, not counting the ones that were requested by multiple sites. Of these roughly 500 genes, the median size was about 1725 base pairs, and that's including the coding sequence only. Generally, when we do these capture panels, we like to pad out the coding region and lay probes into the introns a little bit. So this median gene size, when you consider this padding region, is actually a little bit closer to 3,200. Of these 494 genes, there were 76 genes that were requested by greater than 4 sites, or I should say at least 4 sites, and 2 genes that were overall requested by all 9 sites. Those would be VHL and LY.
Hey, sorry, Adam. Can everyone go on mute so Adam doesn't get the feedback? Thank you.
Sorry about that, folks. So yeah, those 2 genes that were requested by all 9 sites, I think it's safe to say those will probably end up on the platform. On the opposite end of the spectrum, we had 313 genes that were requested by only a single site out of 9. And of these 313, 31 of these are in the top 10 list from at least 1 site. So in addition to a few genes that are requested by almost every site, we have a few that are high priority by only a single site. So in terms of gene size, what are we looking at here? Well, again, this is just on the left, a histogram of the gene sizes for the coding region only, and on the right, including this padding region. These histograms include 493 genes total. This graph does not include Titan. Two sites did request Titan, both in their top 10. And unfortunately, if I were to include Titan on this graph, it would be way out to the side. It's almost an order of magnitude larger than the maximum here. So, including Titan on a platform would take up significant sequencing real estate, and it's something we would have to think about moving forward. In moving forward with the rest of this presentation, Titan has been removed. So this is what you're looking at right here is a 9-way Venn diagram of the requested genes from all 9 sites. It's a little bit hard to read because you can see there's quite a lot of overlap between the sites. And in fact, there are some sites that are completely overlapped by other sites that don't have any unique gene submissions. What you can see here is that everything kind of clusters roughly as we'd expect. CHOP out here, being a pediatric site, had the most different requests from everyone else, although they do overlap a little bit with Partners and Cincinnati, who requested a few carrier status genes and other pediatric type genes. Again, this is more to give you a general sense of the type of overlap in what people are asking for instead of actual numbers. Including numbers on this graph makes it suddenly very, very messy. So moving forward, then what I did is I took all 493 of these genes and I went in and hand annotated with a kind of shorthand phenotype, a very brief description of the phenotype of interest, either from the sites themselves, if I had it, or from OMEM or current literature search or other source. So what you're looking at here is network-wide.
Adam, this is Dan Roden. Can I ask that you spell arrhythmias correctly in the future? Thank you.
Oh my God, did I forget the H? Oh, Dan. All right, yeah, I lose that point. I think it's fixed on all the other ones. Yeah, but anyway, this again, what you're looking at is all of these phenotypes that were requested by the sites. These aren't standard terms, so again, this is just a very broad overview of what people are interested in. A lot of these terms will overlap, and they are not standard, as I said. And we're working on some ideas on how to standardize phenotype terminology eMERGE-wide moving forward, and have some interesting ideas about that. When we break down these site by site, what each site is looking at in terms of these phenotype word clouds, again, we can get a better sense of what each site is interested in looking at. And I'm not going to spend too much detail on going through each one of these for every site because, again, I'm including these more for your reference in this presentation once it goes out to everyone to sort of get a quick glance at each site. Again, here's the next 4 sites. Arrhythmia is spelled correctly here, thankfully. And then Partners is the last one here up on the right. So just to finish off this brief slide set, now that we have all of these gene requests together, one of the things we need to think about is some schema of how we want to take all of these requests and turn them into an actual panel. So in the next week before the PI call, I really hope you guys will all take a look at what everyone is requesting, take a look at the information that's in the matrix, which I'll show you in a second, and really think about some ideas of what this panel might look like. One idea that we had at the coordinating center and the sequencing centers, just to kick off discussion, is a design that centers around the 56 ACMG genes, and then including the top 6 requests from every site. This mock idea would give us 106 genes with a panel size of roughly 400 to 600 kilobases, depending on how much padding there is and how much non-coding space we include for genes. But again, we're very much interested in what the sites have to say in terms of what this panel is going to look like. One other thing that was not included in this analysis here was any of the single nucleotide variants. We did ask sites to submit SNPs of interest that they might want to include on the platform. And I'm working on harmonizing these similarly to how I've done to the genes, although they will take a minority of real estate on the final platform. Most of these SNPs only require 1 or 2 probes, or minimum 2, I should say. So again, they're not going to really be a make it or break it kind of thing for the platform overall. And then finally, in the next week, as more of just a fun note, we're really interested in people's ideas of what we want to call this thing. We've really just been referring to it internally as eMERGE panel or eMERGE sequencing panel. One idea that we came up with was CareSeq, politically associated reasons for eMERGE sequencing. But again, that was just the first thing that popped into people's minds. So if you have more suggestions, we really would like to hear it. So in terms of slides, that's really all I have to say today. So now what I'm going to do is kind of walk everyone on the call through the master gene requests matrix and hopefully take some questions about how you can use this matrix and maybe how it was assembled if people have them.
Adam, can you hear me?
Yes.
Before you move on, I just want to say that people should keep in mind that if genes that they're interested in don't end up in the final 100, they should make sure and take advantage of the opportunity to add SNVs of interest in those genes to the panel if there are particular sites that they're interested in.
Yeah, good point, Gail. And again, I think that's the case. There are several genes that are included basically because there are one or two SNPs that have known clinical relevance. And rather than not including the gene as a whole, it might be preferable to just include those two variants.
Hi, Adam. Can you hear me?
Yes. Yes.
Do you know what the total number of SNPs might be? This is Iftikhar. Or you don't know that for you?
We're not quite sure yet. We're still waiting for the list from every site in terms of SNPs. And we're not entirely sure what that's going to look like and how many we're going to include altogether.
All right. Thanks.
I don't really anticipate having to kick off a large number of them based on what I've looked at so far. So I guess that's a longhand way of saying I would submit more than you think, and then we can cut down later as needed. Okay. So again, now moving on. Hopefully, if you can't see my screen, please speak up. Did someone say something just now? No.
It was me, Adam. I was asking if there were any other questions before you moved over to the matrix.
Okay, cool. So what we're looking at right now is the matrix that I assembled from everybody's gene requests. So just to kind of orient you in terms of what we're looking at here.
Can you maximize the size of that window? Is there a way to make the size of the window bigger?
Oh, I don't actually have a window, so I can't see what you guys are seeing. Is there a formatting problem?
In your window, Adam, if you can make that spreadsheet as big as possible in your window, that would help.
Oh, I see.
Yeah. Just go up to 150, maybe, the percentage up there.
Better. That's better.
There you go.
There. Good? A little bit?
Perfect.
I probably don't see
Okay.
anything, but yeah.
Okay. And again, this matrix will be sent out to everyone after this call, so you will be able to do all of this yourself. There's no way I could summarize everything that's in this table here. There's quite a lot of information here, as you'll see. So this first column here is gene, and what I've done is I've standardized all the genes with their RefSeq names. So if for some reason you're looking for a gene and you don't seem to find it, the first thing that I might suspect is that the name that you're used to calling it might not actually be what it's called in RefSeq. But again, these are the ones that are standard in terms of exome design and UCSC. This is that brief phenotype that I was mentioning before. And again, I highly encourage you, if there is an error here or something that should be clarified, to please let me know. And let me know not by changing it directly in the cell, but instead by making a comment on the cell and then sending it back to me so I can keep track of the versioning and what gets changed between all the sites. So the next column, of course, is which genes are on the ACMG list. And again, here you can, in any of these columns, you can filter by clicking on these arrows here. Here, if we want to see only the ACMG genes, we would just click this X. And there we have all 56. These are two measurements of size that I built the histograms off of before. On the left is the coding region only, and here's with that padding. This information is thanks to partners and LMM, so big thanks to those groups for getting me that. And then finally, you can sort by the total number of requests among all the sites. When we look at that, we sort of get a sense of what, in general, people are requesting and how this matrix tends to look. So each column here are the requests from a different site, and they're all ranked, but they're separated into this orange group and this blue group. The orange group consists of this top 5 and then the plus 5 that was requested by the PIs. And then the blue colors here were the NHGRI requests, which were a top 1 to 100 and a 101 through 125. Now, we did have some sites take a few different approaches to this, and so I tried to keep that represented in this matrix. In general, you can look at what everyone's top list is by simply sorting their column. So, for example, if we were interested in what UW was looking at, we're just going to hit ascending here. And we're going to turn off the ACMG filter. There. And here we can see, so here at UW, what we did was we submitted our top 100, but we also submitted a top 1 through 5 and a 6 through 10, like this. So you can see that represented in this column by these specifically ranked orange cells here, as well as these blue ones, which actually, if you look, do have numbers in them. This one is number 1. This one is number 2. That's purely for sorting purposes. If a cell like this does not have an actual number in it, that means it was submitted to us unranked. So even though this gene is within the top 100, it doesn't really have a relative position relative to the other 100 genes. It's just a member of the top 100. If I did receive an explicit rank from a site, that's represented by a number in the cell, as you can see here. So again, this sort of thing allows you to look at what each site is requesting. Here, looking at CHOP to see that, again, like we saw in the Venn diagram, this pediatric site is really requesting genes that are quite different from the rest of the sites.
Can I interrupt again? And just to remind those of us on the call who aren't aware that in your top 10 list, that was supposed to be exclusive of the ACMG genes. And so there was at least one site that had a couple ACMG genes in there, but we said, what is your top 10, not considering the ACMG genes. So that's why you don't see the ACMGs ranked among the top 10 for people who weren't aware of that. Separate than that, my patient's here, so have a lovely call. I'm going. Bye.
All right, thanks, Gail. And yeah, and I would not certainly treat this as your final ranking. This is not going to be set in stone. I think this is just a way for us to move forward in terms of thinking about the panel, and adjustments will be able to be made to your top 5 or 6 as we move forward. So the last thing that I quickly wanted to go over in this matrix before taking some general questions is how to really interestingly use this phenotype column. And so again, here we're listing, when you click this arrow, every phenotype that I annotated as part of this gene list. What you can do is basically look for sites that are interested across multiple phenotypes. Like if I'm interested in sites that are studying lipids, for example, we can just search for lipids. Anything that's annotated with lipids will come up here. And here we can see that we've got UW and Mayo mostly primarily focused on lipids. If we look at the carrier status genes, we can see that Partners has requested quite a few of them outside of the other ones.
Where was the annotation? I'm sorry. Where was the annotation that was that just grouping based on how people identified them, or how did you categorize?
So again, this is very high level. This is not; these terms aren't formalized at all. Basically, what I did was, if the site sent an annotation with the gene to me, I used what the site gave me. Otherwise, I went and got my own. Okay. Like I said, we're working on a standardized phenotype terminology for this, and kind of more of an ontology style. But again, you'll have to remember, I got these gene lists on Friday night, so
right.
This was a very quick annotation effort on my part.
And so if you do see something that you would like to change or comment, please send it back to Adam, as he indicated. So we're submitting this for help, really. We would love your input.
Absolutely. So again, the more specific information we have per gene, the better.
While we're chatting, would you go through and show the top 10 for each site one at a time, and pause on it for a few seconds, 10 or 20 seconds, so that we can have a sense about what our relationship is with these top choices? Yeah, and actually,
I think rather than top 10, what I'll show you is actually what I was going to show you next, which is that this spreadsheet actually has 2 sheets. If you look down here, the one I've been showing you is this all genes sheet. But the second one here is that kind of design idea that I presented before with this ACMG 56 plus top 56. And if you look here, this is sorted alphabetically, is what you can see here is more or less how each of these sites are related. And if you want, I can show you each site's top 6 briefly. Here for Cincinnati. From CHOP, like I said, CHOP had the least amount of overlap, probably due to their different patient population. Oops. Columbia's is here. Followed by Geisinger. Oh, was someone going to say something?
This is Melissa. Something else that might be useful here that we worked through a little bit this morning is that you can actually filter this to take out the ACMG top 56. So for example, if we were to assume that we would use a large majority of those, once you do that, once you take out the 56, it's actually a much cleaner list of just the sites top 6, as long as they didn't include the ACMG genes. So again, it's just one more way that you can sort and filter and perhaps start to think about this information.
Yeah, and again, I filtered it just as Melissa suggested on the screen right now, and it's a little bit easier to see the orange boxes. So again, I just wanted to give everyone a high-level overview.
But I can hardly read the gene names.
Sorry?
I can see the orange boxes in the blue.
Can you go up to one? Can you go up to one? There you go. Yeah.
But yeah, thanks.
Yeah, so I want everyone to really spend some time with this list on their own and really think about these genes and definitely get to know not only what your site submitted, but what all the other sites submitted as well. I think as a network, it really benefits us to know what the sites have in terms of overlap, in terms of what they have different as we move forward into making the platform and eventually sequencing people on it.
So how would we, just on that point, in looking at what others are doing, and let's say that in seeing that landscape, we want to make some adjustment and swap in a gene or swap out a gene, how would that work? I mean, obviously, we don't want to get into an endless iterative process, but...
Exactly. Yeah. Our goal at the coordinating center is to avoid a kind of endless iteration process. And I think the process of how to go about doing these revisions is exactly what's going to be discussed on the PI's call on Thursday, if I'm not mistaken. And so that's why we wanted to give you guys a week with this list to really think about how much revision would be needed, if any at all, so we could get a sense of how that process can go.
Okay.
Adam, this is Melissa again. So do you not want some changes from the sites even now, as I think that they were saying, hey, looking at this, maybe I didn't send the right thing. Do you not want anything from them now, even before they go into Thursday?
So I will welcome anything that anyone sends, obviously, right now, and I will roll it in. But my intention is anything that I get before the PI's call, I'll probably roll into a version 2 of this file to be released right before that call next Thursday. So I won't be releasing new versions every time I get an update from the site, is what I'm trying to say. But yeah, please feel free to email me.
And on that note, I think it would be great if we could get 1 to 2 individuals or more from each site to participate on a working group for this panel design. So that's something else we would like by next Thursday.
Yeah, so I'm sure as you're taking a look through this file over the coming week, you'll have some questions come up or comments. I encourage you to send me an email. I should get back to you very quickly. I'll send my email out. It's on the listserv, as well as I'll send it out when I send this matrix. The other thing I want to note is later today we have a metrics table coming that has a much more detailed description of each of these genes with a variety of metrics collected by me, as well as help from the sequencing centers in terms of which genes are currently tested at the sequencing centers, some metrics about how well they sequence, and a little bit more detail in terms of phenotype. So I think that'll help think about these genes moving ahead, as not everyone has every single one of these genes memorized. I certainly didn't. So if no one else has any more questions or comments for right now, I will leave you all to digest this, and I encourage you to look for my email containing the Excel sheet in a few minutes.
Well, thank you, Adam, for presenting. Do we have any questions while we have Adam on the phone regarding the matrix itself, or design plans?
So was the Venn diagram you showed earlier before you launched into the individual genes, was that based on the 6, the 10, or the 100 genes?
That was based on the total 125 list. Yeah, all genes were tested.
Adam, would it be useful to do the redo the Venn diagram without the 56 ACMG genes, just to get a sense of?
Yeah, I could do that. That would actually be that would be pretty easy for me to do. I could do that and send that along with the metrics table a bit later today.
And you're going to send out those slides anyway, right?
Yes, absolutely.
Right, great.
This is
And don't take the arrhythmia comment personally. Everybody misspells it. It's just I have. You would be disappointed if I didn't say something. I mean, I just have to. That's.
I'm
going to mute myself now.
I take spelling very seriously. So.
Adam, will you generate a hit list of the 106 genes that you think were chosen, the ones that we would most like to get rid of in that list? The ones that have duplicated pseudogenes or are huge or other problems that
yeah. So again, that kind of stuff is what we're gonna be rolling into the larger metrics table. I got this morning some metrics from the partners in LMM Sequencing Center in terms of mappability, homology, difficulty sequencing that we're planning on putting in there for exactly that reason.
So this is Iftikhar. Can you hear me?
Yes.
So will we be? I think I'm not sure how this process will go forward. Is there a request from the sequencing centers for a certain number of sequence, in terms of base pairs that will be sequenced, or is it the number of genes? And at which point will the actionability annotation be taken into account? Do you know that, Adam? That's still being discussed.
David?
Or David?
Yeah, I'll go ahead and field it, Iftikhar. So I would say that we're really just in the heat of those discussions right now, and the point being, let's see what we want or what we can agree on, and then through negotiations we're going to talk about just how many. I know we keep on hedging towards this ACMG plus the 6 and everything, because that gets us in the ballpark, but that's the whole purpose of the discussion next Thursday.
And will there be sequencing site represented on that call? Or is it just PIs?
Yes.
I'm sure there will be.
No, the sequencing centers will be there.
Okay.
Yeah.
And I'll probably be sitting in, listening in as well, too.
Yes.
Yeah, I will say, if you do have questions, I'm sorry, go ahead.
Well,
I was going to say
no, this is Melissa. I was just going to say Adam would have to be there to do some of, I think, because we're going to be doing some sorting and discussion of this. So we will have the sequence. The sequencing centers are on the PI group anyway, but definitely Adam Gordon would be there as well.
Right. So, is there an expectation of an updated list to be sent before the call, for example, with annotation, particularly regard to actionability, or are we just going to, at this stage, go with what we have collated so far?
So those, the fuller annotation table, you'll actually have later today.
You mean what kind of annotation, Adam?
Sorry, that should have been a little clearer. It's basically a large table with genes. Let's see if I can bring up the draft I have. But it does include metrics on actionability and whether it's tested currently at the sequencing centers.
Okay, well, I'm surprised. I mean, some of these are probably, you need input from content experts to determine actionability. So where is that annotation coming from as far as actionability?
Right. Well,
I believe, Adam, correct me if I'm wrong, but this is coming, a lot of this is coming from Heidi's group that works, obviously with the ClinGen.
Okay. Okay,
so,
well that's good to know.
Yeah.
Yeah.
My answer would have been ClinGen.
Yeah.
Yeah, we had to start somewhere, so that was as good a place as any, we felt.
But we absolutely want input, and so I would say that don't wait, Iftikhar, if you have something that you would like to bring up, do not wait till next Thursday. I mean, go ahead and start sending emails. This is just really to initiate that process.
Okay, well, what we'll do is we'll wait for the annotated file and we'll review it, and then if you have any comments, we'll send those before the call.
Excellent. Yeah, and if you can if you're looking at the screen again, this is sort of what the what kind of information is going to be in that metrics table once it gets sent out later today.
Okay, and there's an actionability column. Okay, good.
Any more questions?
Okay, yeah. Any more questions for Adam? And I would just like to also thank Adam for putting this matrix together so quickly. As you know, we received the list last Friday, and he had this matrix put together by Monday. So things are moving quickly because of him, and I think that this process is going smoothly.
It is reassuring that out of 9 sites that each have 125 genes, that we don't have 1,100 separate gene nominations. So the Venn diagram just shows that most great minds, at least for adult medicine, think alike. I don't know about the kids. That's a separate.
Right, I agree. I think in total we're going to end up with a really, really strong panel that's going to give us some really interesting results across all the sites.
Okay, thanks for the comment, Dan. And so we all have homework. Please think about the work group nominations. If to send a couple of individuals that you or yourself for that, and do not forget about the SNPs. Please send those in as needed. So, are there any other questions or comments?
In some degree, the SNPs that we would pick would depend on which genes get picked too, though. Probably. So they might have to go in parallel, I guess, or one after the other.
That's very true, Josh.
Right.
Yeah, it might be a 1-2 kind of thing. So if certain genes are do not get selected, it may impact which SNPs are selected based on the science might change a little bit.
The other thing I was thinking about was the so Adam, you said that if you include 60 extra genes based on our selections, you ended up with 106 genes. Is that right?
Yeah.
So there was enough overlap. The top 6, sorry, I should say, resulted in enough overlap that there was some combining. I wonder if there were candidates that maybe we ranked 10th, but nearly all of us ranked it 10th that we need to look at. Did that occur?
Ranked at? I'm sorry, I couldn't hear what you say.
Were there genes that you said you included the top 6 in that initial stab, but are there genes that maybe we ranked in the top 10, or that didn't quite, that maybe all of us selected, or a lot of us selected?
Right. Looking at what's in 7 through 10, basically.
Yeah.
Right. I don't think so, Josh. Adam can put that back up there, or you can sort when you get the thing. There wasn't quite as much overlap as you might think.
Right, and the other thing, again, this is just a first pass because I think, again, we got a number of top 10s or top 6s that were unranked. They were just a list of 10 genes. So moving forward, we're going to need a very explicit, 1 through 6 ranking from folks, I believe, just so I don't end up arbitrarily picking who's number 6 based on who was first in the list.
Okay. Are there any other questions or comment? Well, thanks for everyone calling in. Adam, thanks for presenting. And, we will talk next Thursday.
All right. Thanks.
Thank you
Great
all.
work. Thank you all.